Now, turning our attention to Postdoctoral Researcher, the results are actually quite interesting and a little surprising as well. So the same things are plotted here on the X and Y axes, and biofilm is used as a natural inducer, and cesium chloride, once again, is used as an artificial inducer. So I'll walk you through this. So looking at just the black bars first, the biofilm inducer, we see that it induces whole larvae to settle and metamorphose, a very high percentage of them to do so, in fact. Among the aboral ends here, it actually did not induce the quarter aboral section to settle or metamorphose. And it did induce the 1/2 and 3/4 aboral ends to metamorphose. And what's really surprising is this section here. So this is 3/4 oral. That means it's missing that aboral end. It's missing that aboral tip where we think that potential sensory cells are localized. But missing it, it's still able to metamorphose. So what is going on here? And this is something that I will address in the next slide, really. And going back to this artificial inducer here, I want to walk you through that as well, because it's pretty important. The artificial inducer induced whole larvae to metamorphose, and it induced a lot of the aboral sections to metamorphose, as well as these oral sections. It did not induce this last quarter here. And so once again, with the cesium chloride, this suggests that these sections have not lost their capability to undergo metamorphosis. And that is particularly true about this one. So this is really interesting in that it did not get induced by a natural cue, but it did get induced by an artificial cue. If we are to look at the schematic once again and think about a larva in 4 different parts, and go back to the graph and compare, we can see that this has a part 1, which did not get induced, and this section has parts 1 and 2. This type of section has parts 1, 2, and 3, and this type of section has parts 2, 3, and 4. And what's common about these that got induced by the natural cue is that they all have a part 2. And so this suggests that there may be potential sensory cells that are localized in this particular area. And part 1 specifically has been shown not to be induced by a natural cue. And so what this might indicate is this species is sensing biofilms on their sides, really. So instead of using their aboral end to detect the cue, even though that's the end that eventually attaches, the sensory cells could be localized along the sides of the larvae. And later on, I had gone on to confirm this with some more behavioral studies as well. So in getting back to these questions here, which region of the larval body is directly involved in the detection of cues? It is the aboral end, but it's dependent on the species as far as which part of the aboral end. And so for Montifera capitata, it was that first aboral quarter, and for Postdoctoral Researcher, it was that second aboral quarter. And if that region was removed, would larvae still be able to undergo settlement and metamorphosis? And the answer is no. So even though, going back to the big picture, even though I was not able to actually manipulate individual sensory cells, this is an abstract way to kind of determine which region sensory cells might be localized in by doing these types of experiments. And in case you're curious as far as what sectioned larvae look like when they settle, I followed them through. So this is post-settlement development, and these were followed through 3 to 7 days after they had settled. So just to show you here, a whole larva, not cut, looks like this when they settle, and this is a normal phenotype. And when you have the oral end missing, it forms an abnormal mouth, as you would expect. And when you have the aboral end missing, it forms an abnormal base. So it's lacking this nice fringing pattern that you see in the normal phenotype. If you are to follow this for a month, then you see a clear, distinct delay in development. And whole larvae by this time have developed multiple polyps now, and oral with the sections with the oral ends missing have only a single polyp, and even the tentacle buds at this point are short and stubby. And the aboral end missing only has a single polyp at this point too. So this suggests that regeneration in the species is very slow. So in summarizing this part, my dissertation research and what I have found when I looked at both the cues and sensory systems, the sources of these cues for settlement for a lot of marine invertebrate species vary widely, and bacterial biofilms appear to supply the cues for a lot of marine species. Species of the gamma proteobacterial genus Pseudoultramonas, as I've already mentioned, have been shown to be strong inducers for settlement in various marine invertebrate larvae. So that includes not just the coral larvae, but a sea urchin, a bile-following tube worm as well. And the distribution of sensory cells for chemoreception varies across different invertebrate phyla. So it's not all the same. I mean, at first I thought, okay, Montripora is a coral, Postdoctoral is a coral, what can possibly be different between the two? And what my results show, or suggest at least, is that the localization of sensory cells are different in two different species. And who knows if that is due to their different sizes. I think that could be a huge issue, that the Postdoctoral damacornis larvae are much larger, or that they have different reproductive strategies. And this kind of dives into some future work, and as far as what future directions this field can go. A complete bacterial genome, for example, one for Pseudoultramonas luteoviolacea, would be especially useful because it is a strong inducer, and it induces a lot of different types of marine invertebrate larvae to settle and metamorphose. And so a genome would allow a deeper investigation of the molecular components that produce the settlement signal, which is really needed at this point, as we still don't know what it is about bacteria that make them inductive. And finally, looking at the animals now, complete transcriptomes and genomes of more invertebrate larval species would provide a means to search for candidate receptor genes and their different functions, and what could be involved at the molecular level in terms of sensing cues. And this kind of ties into what I'm doing for my postdoc work right now. So I'll talk a little